Today we are fortunate to have Professor Charron, Dominique Charron, with us. And Dominique studied medicine and did his PhD at the Paris University. And he moved to do a postdoctoral fellowship in the Stanford University, I believe. For how long, Dominique? You stay?
4 years.
4 years in total. That's a long time. So he came back to Paris to take the chair of the Department of Hematology and Immunology and Therapeutic Targets at the Hôpital Saint-Louis at Paris. Apart from this, he's president of several scientific organizations, and he as well is acting as an advisory expert in different bodies like the European Union, the UNESCO, among others. So big experience publishing quite a lot of papers in the high-impact journals. Today he is going to talk about his work, Allogeneicity and Immunogenicity of stem cell therapy, particularly focused on the cardiovascular system. Okay, Dominique.
I'm trying to think of immunology in the perspective of use what immunology and transplantation immunology can bring to the cardiology community in terms of regenerative medicine. So that's what I would like to talk about today and give some perspective and some thought and also present some recent data. So some of the focus will be on cardiology, but it will not be only on cardiology. So basically, this slide is just to remind you that immunogenetics and medicine has been a very successful story in the 20th century. The HLA was discovered, the Hla system, and that has allowed to perform transplantation. It has also an impact on genetic susceptibility, prediction of disease, autoimmune disease. And of course, the HLA system is what controls infection. Well, in the 21st century we are in, there is new aspects of HLA, of the HLA system that have been and are being discovered. First, many diseases and even psychiatric disease or neuropsychiatric disease have a very heavy component, genetic component within the major histocompatibility complex. I'm not going to talk about that at a cardiology meeting. I only talk about that at a psychiatry meeting. There is also this field which has been developing fast where susceptibility to drug response, including negative detrimental response to some antiviral drugs or some other drugs you know, are under HLA control. And to the point that now you do HLA typing before you prescribe some of those drugs to try to prevent and actually succeed in preventing those accidents. And then there is this field of cell stem regenerative medicine that we will talk about. First, I just wanted for some of you that may not be totally familiar with the HLA system to give you a glance at the complexity. But don't be afraid of the complexity because we can take care of the complexity now. This system was completely sequenced and mapped, as you say, by an international cooperation at the end of the previous century. The major issue are actually what are called the transplantation antigen, the classical HLA, class 1, HLA ABC, or class 2, HLA DR, DQ, DP. So that's the class 1 major histocompatibility complex antigen and the class 2, and those are what basically prevents transplantation. If we look at the diversity now, not the number of genes, which was on this slide. But now if we take individuals and we look at the genetic polymorphism in the population, this is kind of frightening slide. And you see, this is a time where it wasn't that long ago. There were slightly over 1,000 genes, alleles of the HLA system in 2000, and now we are over 8,000. I just want to put this slide because I'm head of the Jean Dosset laboratory who discovered the HLA system and was awarded the Nobel Prize for this discovery. Here is Jean Dosset with the first HLA antigen. He called it Mac. So it was not Macintosh, but and now, in 2013, I checked a few weeks ago, we are over 8,000 allele. And when I say we are the limit of this diversity, the diversity of our biological self, and this is not the combinatorial diversity, this is the individual allele diversity. We are the limit because there are many populations that have not reached the numbers that is actually expected to. For example, in Africa, we have quite a low representation of the alleles. There are many more alleles to discover in that situation. So those alleles, those HLA antigens, HLA class 1 and HLA class 2, they define the biological self. That means when you are in a situation of transplantation, taking an individual, either taking the cells from an individual, taking the organs from the individual, taking the bone marrow cells from the individual, taking tissue. Those are all settings of transplantation. They will be recognized, those antigens, class 1 and class 2, those transplantation antigens. That's why they were called. And they will be actually eliciting an immune response. That's called allo-recognition and allo-immune response. And that will lead ultimately to rejection. The reason why I show this slide is that there is two pathways for this allo-recognition. Allo-recognition is actually between individuals from the same species, the recognition of the MHC. You have what's called the direct allo-recognition pathway, where the class 1 and class 2 are directly recognized by the recipient. So these are the donor cells. Those are the recipient cells of an organ, for example, and the T cell from this donor will recognize from this recipient will recognize the donor MHC molecule and develop a T cell response. But there is also an indirect recognition pathway, which is also an important one. In that case, the cells that differ from the donor and the recipient, and the molecule, the HLA molecule from the donor, will actually be taken up by an antigen-presented cell. They will be processed by that cells from the recipient, and they will be put at the surface of that cell, the recipient cell, as a peptide. But this peptide being different, it will be recognized also by the recipient. So this is an allo-recognition system that is different in terms of not the molecules that are recognized and that elicit the system. This is class 1 or class 2 differences. But the type of response, for example, in the case of the direct, this is a pre-existing situation. And we all have T cells at a quite high frequency in our body that if we got a transfusion of a tissue bearing class 1 or class 2 molecule, those T cells are specific and will react against those cells and will destroy those cells. So this is an immediate response which pre-exists, which is at high frequency, and which induce mostly cytotoxicity and cytokine production. The duration of the response is short-lived because after you have the cells that are present, they kind of disappear. And this elicits mostly what's called acute rejection. And that's why you cannot do a transplantation without pre-treatment. The other system is interesting because it's mostly a B-cell activation system. And in that case, it's the other arm of the immune response which develop. Of course, the T cells are the helper T cells, but you have antibody production, and that's mostly anti-HLA antibody production that elicits this type of allogenic response and was thought to be responsible and is responsible for chronic rejection. But you will see that if we take the consequences of HLA histocompatibility in transplantation, whether it's organ transplantation, whether it's bone marrow transplantation, the T-cell mediated, T lymphocyte mediated aspect is a rejection for organs or the GVH. And it was considered really as the major rejection phenomenon. The anti-HLA antibody that developed later on were thought to be responsible from the chronic rejection. However, very recently, and we have shown that in the lab also, in terms of cord blood transplantation, a particular case of hematopoietic stem cell transplantation, you have no engraftment. So this is an issue which is very similar to cell therapy, any type of cell therapy. But in 2013, actually the end of 2012, the change of paradigm in that issue will come, and I will discuss that with you. So the consequences of mismatching, this is just a summary slide, in renal transplantation. What you can see here is that depending on the mismatching at the HLA-A, B, or DR loci, you have zero mismatching, a better graft survival than 6 mismatching. And this is on very large number of patients. This is an international European cooperation. But what we were surprised to observe in organ transplantation, and this is renal transplantation, but it may apply to actually also heart transplantation. And some of you are heart surgeons, some of you may be interested in heart transplant. And so the issue of antibody-mediated rejection, rather acute rejection, is an important issue. And the dogma for a long time was, still is, but it's going to change, is that most of those rejection, and even not all of these rejection, were T-cell mediated. That's the reason why the treatment for those organ rejection, kidney rejection, are actually immunosuppressive directed at T cells. Well, we wanted to revise this type of study, and we embarked in a very large population-based study of over 2,000 patients from our two institutions, St. Louis Hospital and Necker Hospital, and for which we had proven rejection on over 300 biopsies. So this is 10 years, a very large clinical study, where we had the clinical data, the histopathological data, including the deposition of the C4D, and the presence of donor-specific anti-HLA antibody. And we were able to define 4 patterns of rejection. Not one pattern of rejection, but 4 patterns of rejection. And basically, the classical T-cell mediated rejection with T-cell infiltration, the antibody-mediated rejection, and also this case, but with a big difference, is that we added the look at the vasculitis. What is the situation of the endothelium in those organs? So you have those categories with vasculitis or without vasculitis. And this is, that slide went off. I don't understand what happened, but that's not a problem. This is not a Mac compatibility. The other slides are okay. So this was just to show you in those different categories the different four type of phenotype of those four rejection patterns. So overall now, if you look at organ transplantation, and this is demonstrated for kidney transplantation. This is this Lancet paper, and the study is being performed now for other type of organ transplantation. And the reason why I'm showing you that is that I think it will apply ultimately to stem cell transplantation. We have a new category of rejection here, which is anti-HLA mediated rejection with vasculitis. And the reason to define this is that, but again, there's a problem here, because what that slide was supposed to show is that if we look at the result of the survival, this is the worst case to predict rejection. This is the presence of anti-HLA antibody mediated with vasculitis. So that's basically, this were survival curves, and those survival curves show that you have those different categories of rejection, and those different categories of rejection identify this new pattern of rejection with a very bad prognosis. And also, what is very important in that case, and if we can predict those type of rejection, those cases are not susceptible to be treated and to respond to anti-T cell immunotherapy. And that's the main, actually, issue. So that was just to illustrate the bridge that there is from the transplantation histocompatibility and the transplantation field that has been a success for 50 years, and what we know about how to deal with those patients with what is now the future and you feel, which is to use a series of stem cells for regenerative medicine. So as I said, the field has really started in cardiology and also somehow in neurology. And if we take the stem cell therapy for regenerative medicine, I'm not going to detail their benefit because they are well known from all of you. And that's why those cells are really very good candidates for restoring actually the function of ailing organ. But there is a question that was raised very early on from embryonic stem cells, for example, or from other type of cells. This is whether those cells are immunoprivileged. And I put a question mark. It was not a question mark many years ago. It was kind of a dogma. And now it's not only a question mark. I think I will show you we can write off this question mark. They're immunospecific, but they may not be immunoprivileged. So clearly the limits of the in vivo engraftment and functionality of those cells in regenerative medicine are the disponibility, the timeline, the aging, the safety, all those issues that you all know well, but also this immunogenicity and allogenicity we should consider. So I just want to review some data from the literature before going into my own lab data. First, what are the immunity factor in regenerative cell therapy? Well, first you have the immunogenetic factor. You have the HLA expression, the MHC, and the MHC is a major histocompatibility complex, but there are also other systems that are called minor, although that means that once a major is matched, then you can uncover new histocompatibility system. Then you have the immune effector cells with the direct and the indirect pathway of allorecognition, and then the aging factors that I don't want to talk about today, but which also is an issue because there is a phenomenon which is called immunosenescence that can take place. So what we would like to do in the future is to have really a choice of stem cells that is based on our knowledge on their immune behavior. So the allogenic stem cells are not really immunoprivileged because those 3 points, and there is supporting paper. They express MHC, their immunogenicity increase upon differentiation, and you can have even demonstrated in vivo rejection. Very briefly, this is the data on embryonic stem cells, where you can see that during differentiation, actually, they have low level of HLA class 1, and they will increase their level of HLA class 1 in their differentiated states. And this is already 10 years ago, there was evidence that the MHC was expressed on those cells. And if you use interferon gamma in this case, you can see that you increase the expression of the MHC class 1 in the time and hour and dose-dependent manner. So that means that those cells, embryonic stem cells or cells of this type of stem cells, they will react very similarly to any other cell where they are subjected to interferon treatment. This cannot be seen very well, but it just shows that when you use those embryonic stem cells in vivo, after transplantation into an ischemic myocardium, this is a work you probably know very well, you can have actually, if you use syngeneic, so autologous, like, or allogenic, if they are allogenic, then you have an infiltration with T cells and an infiltration with B cells in the myocardium. So they elicit an immune response in the site of injection when they are allogenic cells, while when they are syngeneic cells, they don't. And finally, there was this work presented in Madrid a few years ago that shows using mesenchymal stem cells that during the myocardial repair, and it's a rat model, you could say that while those cells, when they are in the stem cell stage, have a low immunogenicity, they become immunogenic in vivo and in vitro and in vivo, and actually you have rejection of those cells. And even in that case, you can lose the restoration of the cardiac function that was shown. So those data and coming to a cardiology meeting and meeting Manolo and meeting actually also Bernardo Nadal-Gina, that's actually got me into willing to study this kind of problem. So I just want to go into my cardiovascular focus and study the allogenicity and immunogenicity. So we collaborated within European FP7 program carried out with the group of Madrid that has provided us with human cardiac stem cell and stem progenitor, because there is a discussion whether those cells are totally stem or whether they are a mixture of cell or progenitors. But for the immune purpose, that's not a major issue. So when we took those cells are obtained from endomyocardic biopsy. They are after collagenase treatment and c-kit purification. Here you have the pluripotency transcription factor that show you that they are bona fide actually stem cells, and they can differentiate in vitro in cardiomyocyte, endothelial cell, and smooth muscle cell. They have stem cell markers. I'm not going into details. It's published in our paper, and they actually keep their cardiac lineage stem cell marker even under differentiation. So they are very similar to what Bernardo had published years ago. So when we took the cells, we wanted to look at their behavior and their immunogenicity profile. And first, interestingly, those cells grow under hypoxic condition, which is an important issue for the future and which may have some epigenetic effect. And we wanted to take those cells in their resting stage, non-inflammatory or inflammatory. We could actually confirm that even after 72 hours treatment with interferon gamma, they keep their pluripotency transcription factor signature. And so when we look at the expression of the MHC class 1, class 2, and also the co-stimulatory molecules, you can see that those cells do express, and they, under interferon, express even more the HLA class 1 molecule. They are class 2 negative, and under interferon gamma, they express class 2. For the regular co-stimulatory ligands or receptor, they are not positive and not inducible. But interestingly, we found that for one of those co-stimulatory factors of the T-cell activation, I just want to remind you that T-cell activation goes in two stages. The first phase is the recognition by the T-cell receptor of the MHC and of the peptide presented. That's the first signal. And then there is a co-stimulatory signal which is engaged between B7, for example, and CD40 ligand, and also PDL1, PD1 molecule. So this is an important co-stimulatory. All the other one, we didn't find any change. CD80, CD86 didn't change in expression profile, but PDL1 was already positive and increased. When we look at the capacity of the cells to induce in vitro an allogenic response, which is a T-cell response, this is shown here, and this is compared to the allogenic peripheral blood mononuclear cells, which is the regular cells we use in an allogenic response. We could see that there was no great, production of, there was no great expansion of CD4, CD8, but some, with little production of interferon gamma or interleukin 2. But there is actually a production of interleukin 10 by those cells. So this is also an important point here. So basically, those cells have a low phenotype in terms of immunogenicity, and they have a rather low capacity of inducing T-cell proliferation, but they do induce some T-cell proliferation. The second thing we wanted to investigate was not their capacity to induce allogenic response. We had shown they were inducing a low allogenic response, but whether they were also able to modulate an allogenic response or an ongoing immune response of another type that from a third party. And so we were able to show that those cells, and it's shown here, those human cardioprogenitor stem cells can downregulate an ongoing immune response. And this can be shown at the CD4, CD8, interferon gamma, interleukin 2. Those are the detail of this study is in the published paper. So what you can see here, for example, if we stimulate those cells with PHA, so that's the proliferation of those cells, when we use those stem cells, you can see, and whether they are with interferon or without interferon, and we compare that with a kind of standard in modulating an ongoing immune response, which are human mesenchymal stem cell, you have a decrease of proliferation. And this is here linked to an increased production of actually interleukin 10. And that's an important issue that you can see that those HPC, similarly to the human mesenchymal cells, have actually a stimulation of interleukin 10 production. So the ratio of interleukin 10 and interferon gamma is an important one, and you can see that this ratio is very high for actually the human cardiac progenitor cells under inflammation situation mimicked by the interferon gamma. So this actually immunomodulation question, what was actually the cells that were induced to regulate this immune response. And so the question is basically the identification of the type of cells that those human cardiac stem cells will induce. And as shown here, and it was shown before, as I said, there is little activation and expansion of regular CD4 and CD8, and no real change in the Th1, Th2 profile. But when we look at those cells, CD4 plus, CD25 plus, FOXP3, which are actually the profile and phenotype of regulatory T cells, we can see that those cells are able to specifically induce, whether they are resting ones or stimulated with interferon, those type of cells. And this is illustrated here. So from this type of data, we were able to show that those allogenic, and it was done on 3 types of allogenic cardiac stem cells, to activate and expand T regulatory cells based on this CD4, CD25, FOXP3 phenotype. Interestingly, when we look at those cells, and also because we had shown previously the induction of the actually PD-1 ligand, we were able to show that those Treg cells, that actually are characterized by this regulatory phenotype. In addition, are HLA-DR positive and PD-1 positive. So this is the characterization of those cells that are specifically activated and induced to proliferate by those stem cells from cardiac endomyocardial origin. This is shown here under a stimulation condition. So this led us to consider that there is a type of, very specific type of regulatory T cells that are specifically induced by those endomyocardial cells. And those are different from the ones that are induced by the mesenchymal stem cells. That may actually show already and suggest that different cell type, stem cells, in their immunomodulation potency may use different cell types. They may induce different regulatory T cells and may actually use also different molecular pathway. In order to confirm that those cells, those Treg, PD-1 cells were the one responsible for this immunomodulatory. I didn't drink wine, so that's maybe it's the snow from Paris that's getting at me. You wanted to put some cognac in my coffee, so probably you did when I was. Okay, so we use actually two straightforward approach to prove that point. An anti-PD-L1 antibody, and what you can see here, that when you use the anti-PD-L1 antibody, you actually abrogate partially the actually done immune regulation. And so this is both with the resting cardiac stem cell and the interferon-treating cardiac stem cell. And we use also specific scRNA inhibition of the same molecule, PD-L1 siRNA, with all the control, and we got the same result. So not only we were able to show that those cardiac stem cells were able to specifically induce a subpopulation of regulatory T cells, but also those regulatory T cells were acting in immunomodulation through a PD-L1, PD-1 pathway interaction. And this is something that was new in that case. We were also able previously to show that if you use a Transwell system, you don't have this phenomenon. So you needed the interaction, a direct interaction of the PD-L1 ligand on the cardiac stem cell and the PD-1 molecule on the regulatory T cells to actually activate and immunomodulate the ongoing immune response. So in conclusion, from this piece of work, we can propose that those cardiac progenitor stem cell or cardiac progenitor stem cell population, and interestingly, the results are not exactly the same in terms of quantity of action from different donors, but they are all the same way. They are quite attractive for clinical translation because they have a low immunogenic profile. So that means that they will not elicit a very strong immune response at the beginning. The immune response I mentioned to you, the T cell allogenic activation, will be probably not very strong, although it will be there. Then the other point is that when those cells differentiate and when those cells are under and within inflammatory condition, which in the case of myocardial infarction is the case, this immunologic property of immunomodulation remains and is still active. So this is the first point. The second point is that this pathway, PD-L1, PD-1, is the one that is at the origin of the generation of the Treg and of the cardiac stem cell induced modulation. So we have now a target eventually for even therapeutical intervention in that case. So that leads us to this question, which is, beside the detrimental effect we can all consider in terms of injecting an allogenic cell, with the risk of that cell being rejected and inducing actually inflammation, there may be also another side of the coin. Those allogenic cells, because they are allogenic, could be actually beneficial to the clinical situation, because they bias the immune response towards the Treg, and because they are controlled by this pathway. So now to take this overall concept of allogenicity, and I just want to say that this is not news that allogenicity has a janus behavior. On one side it's beneficial, on one side it's detrimental. I always give this data that in bone marrow transplantation, it is the same allogenic reaction that can lead to graft versus host reaction or graft versus leukemia. And it's a balance between the two. And the survival of the patient needs that balance. So you have a beneficial effect and a detrimental effect at the same time. And they cannot be really separated. They can be balanced. So really, in that case, we may be in the same situation, and we should consider the allogenic-driven risk versus the allogenic-driven benefit. And I will propose that we should have those criteria that are listed here that should be studied in order to qualify those allogenic cells for the power of being beneficial. And if we are able to work on those markers and to try to define different stem cells, different allogenic stem cells, in some cases it could be even an autologous stem cell of different origin. It can apply to all the stem cells. If we can actually have this type of characterization, and there are other means to characterize those cells who are ongoing at the moment, including some proteomics studies, then we could be able to do what was the initial goal, an immunologically educated choice of allogenic stem cells. And the second point is that eventually, having some target like the PD-1, PD-L1 pathway, we could even optimize the allogenic stem cell for the use of in regenerative medicine. So this is that data I wanted to present. And I just want to finish also to tell you that beside this very new concept in immunomodulation by stem cells of a regenerative medicine therapy, there is also some points that can be considered from an immunological point of view. I showed to you before that the allogenic embryonic stem cells are immunogenic. I just mentioned that the iPS are also immunogenic, inducing autoimmunity. That was a paper published 2 years ago. A few weeks ago, a paper in Nature said they are not immunogenic. Well, I believe those two papers, which means that if you use those cells, in any case, you will have to prove that the cells you use are not immunogenic. There may be some situations that some of those iPS cells are not immunogenic. There are situations where they are immunogenic. And besides the fact that all those data were mouse data, and they cannot be used for predicting the human data. And all the other situations, we mentioned that also any cell which is allogenic, or any cell which is transformed, have the potential to induce an immune response. And so this is not a dogma, this is reality. And so we should not ignore this reality. We should not try to figure out that those cells are not immunogenic. They are immunogenic. We can try to engineer the cells, or to engineer, in the case of gene therapy, the vector, to have them very weakly immunogenic, but we should not ignore they are immunogenic. But there are solutions even if they are allogenic or immunogenic. And the selection of the cell is one. That's what we do all the time in organ or bone marrow transplantation. We select the best cells for matching. So we can have banks of stem cells can be developed. We can, and I'm not going to talk about here, but there are many means of inducing tolerance. But there are some means that are used in clinical transplantation for over 60 years, and it's called immunomodulation. And immunomodulation works very well. So immunosuppression, as we could say. And that can be used. So I just want to say that there is a field that is developing now in terms of stem cell for regenerative medicine. And this field is to have some stem cell banks, which could be from autologous cells, and this is an anticipatory action. And this is mostly to avoid the aging situation. I don't have time to go into detail here. But if you have autologous cells that are cryopreserved many years before they are needed, then they avoid some of the aging aspect. You could have cells derived from homozygous individuals. You can use frequent haplotype, or you could use allogenic cells. In any case, if you take those cells from the stem cell banking system, when you perform the regenerative medicine cell therapy, you can reduce the HLA mismatching. You can, by this aspect, have the HLA matching, and you can also check the patient for anti-HLA antibody and do cross-matching. This is very important because in the case of cardiac, actually regenerative medicine, some of those patients will actually have and could be eligible also for cardiac transplantation. And you don't want to jeopardize their future by inducing anti-HLA antibodies that will be detrimental for them when they undergo cardiac transplantation. So I just want to finalize in a very pragmatic way, and this is now more transplant immunologist point of view, and this is an everyday aspect that we can already use. Minimizing the immunogenic differences is feasible, and it's done in organ and bone marrow transplantation all the time. Assess immunogenicity status of the recipient prior to and after injection, monitor the allogenic and autoimmunity. And this is the type of protocol we should be developing in CareMI, for example, in the future. But also, before we are sure that we can actually balance the beneficial or the detrimental effect of allogenicity, if we want the cells to stay long enough to do some repair effect, positive repair effect, we can be very pragmatic and actually use the classical immunosuppression protocol for a while that can be translated from organ transplantation, for example. So that's all what I wanted to say, and I just want to acknowledge the work was done in Hôpital Saint-Louis in Paris here, and with my colleague Rimal Dakak and postdoc students involved here. Those are the people from the clinical laboratory that did the phenotyping and the HLA typing. And this is a collaboration with colleagues in Madrid from Core Therapies and with Bernardo Nadal-Ginar and Georgina Ellison. And this is mostly done under this FP7 KRMI consortium. So just the end. It's not the end. It's not even the beginning of the end. It's perhaps the end of the beginning. Thank you.